Hi, everyone. Welcome to SAS Global Forum 2021. We hope you're enjoying yourself. And Yuting and myself, who will be presenting this paper, are also, we've had a lot of fun, both in terms of creating this paper, in terms of doing the research for this paper, and finally presenting this paper at SAS Global Forum 2021. So we're thrilled to be here. And we definitely appreciate everyone's attention as we move on with this paper and this conference in general. We know it's been a tough couple of years, so we appreciate everyone that's attending SAS Global Forum, and of course our ability and our luck to be able to present this year. So the title of our paper is called Generating XPT Files with SAS, R, and Python. And before I go into the paper, just a quick intro. We don't have the luxury of doing this with the co-chairs since it is being recorded. So I'm currently a senior director at Vertex Pharmaceuticals in the stat programming department. I also lead the renal disease area, data standards, strategic outsourcing, and something we call QA, which is kind of a third level of quality in addition to production and QC programming. I've previously led regulatory teams to filings at other companies, and I've also been a pretty regular presenter at Fuse, SAS Global Forum, PharmaSci, and single day events, both in PharmaSci and Fuse as well. So I probably, if I was in an audience, I would probably know some of you, but it's great to be online, and it's great to be presenting. And I'd also like to introduce my colleague and co-author, she's also presented. I actually met her and we interviewed her after a conference a couple of years ago, and she joined Vertex. So we're really thrilled to have her. And I'm personally thrilled to have her working on this paper with me. She's interested in exploring research and statistics. She's currently working as a senior statistical programmer, as I mentioned, at Vertex. And she's also presented, as I mentioned, with SAS and R in SAS Global Forum 2019, JSM 2020, as well as local SAS user conferences as well. So a little bit more about our paper. So our paper is based on a paper that's generated from the FDA itself. The FDA presented this paper in November 2019, and it was called Creating Simplified TS.XPT Files using R and Python. So there was no mention of SAS. Since this is a SAS conference, we are going to include SAS, and we are, at the very end of the presentation, going to make a comparison of the XPT files between R, SAS, and Python. And during that process, Yuting is going to walk you in detail exactly through how to create those XPT files using all three of the softwares. So I think this paper should help address that FDA paper, and we're thinking, that the FDA released that paper for a reason, and that was so that sponsors and other companies could start thinking about using other programming languages in order to create the e-submission deliverables. Of course, we cannot read the FDA's mind on this, but the fact that they released this paper, it's from the FDA, and it specifically talks about R and Python. It actually doesn't talk about SAS in that paper. We talk about SAS here, again, since it's SAS Global Forum, and we're all used to creating XPT files in SAS. That's the default. So we hope you'll learn a lot from this paper. We hope even more that you have a lot of questions from this paper. And just moving along, just to reiterate, the purpose of this paper is to lay out a process of generating a simplified transport XPT file using SAS, R, and Python. And again, we're going to compare these XPT transport files created from the three different languages. And so this is the last slide of the introduction, and then I'll be turning it over to Yuting after this slide, and she'll walk us through the three different scenarios of creating XPT files using both SAS, R, and Python. So what we're going to first do is we're going to use SAS, and we're going to export ts.sas with XPT format, and we're going to show you a macro that we use to create all XPT. So for example, all XPT files in a specific library or directory with SAS. Using RStudio to generate final data sets in XPT format. So you'll see a theme here, right? We're going to generate those final data sets in XPT format with all three programming languages. And we're going to show also with R. We're going to also show a fast way to create all .xpt files with RStudio. Moving on to Python, we're going to also generate final data sets in XPT format. So you'll see a lot of consistency in terms of what we produce in this paper. And you'll see as we go along, with Python, we weren't able to actually generate all of the data sets in a directory. And we're hoping that people would use this paper, use Yuting's research specifically with R, and maybe be able to apply that with Python as well. And for those of you who are using Python to create all of your XPT files, please feel free to get in touch with us. We'd love to hear more about that as well. Lastly, we're going to review and compare the generated TS.XPT files from the three different programming languages. We're just going to open those up in SAS Universal Viewer. So again, we're going to use SAS Universal Viewer to open up the XPT files that we generated using SAS, R, and Python. So with that, I'm going to turn it over to my colleague and co-author, Yuting Tian, and she's going to walk us through the three separate ways to create the XPT files. So Yuting, thank you very much, and I will turn the time over to you.
Thanks, Todd. Hi, everyone. I'm Yuting. So I'm very pleased to present in the SAS Global Forum 2021 with Todd case. And from this slide, I will start to talk about the trial summary domain and the process of how to generate XPT files with three programming languages. So first, at here, you see the trial summary domain is a trial level data set, and each record contains a value of a parameter or characteristic of the trial. And it also records a basic information about the study, such as a protocol title and trial phase, etc. It contains both planned and actual aspects of the trial. We know the purpose of this paper is to introduce the process of generating XPT files with three different languages in terms of original paper created Simplified TS.XPT file. So we assume the readers already have some fundamental knowledge of trial summary domain. So we won't spend too much time in explaining the theory of the trial summary domain. Next slide, please. So this slide shows us how to create the raw TS dataset in SAS. So at here, we say we created the 11 standard variables, which in corresponding with CDISC documents. So this fabricated dataset shows some parameters that we required or expected in the trial summary dataset. So we won't spend much time to talk about how to use data step combined with input statements to create the raw dataset in SAS. But if you are interested in that, you can copy this code and paste in your local SAS software to play with yourself to see what the output will be looks like. Next slide, please. So this slide is output of the code from the former slide. And here we say we have 11 variables. And study ID is 001, the domain is TS. And we set two drugs, drug 1 and drug 2. And TS_parm_CD variable is a short name of the parameter, such as ACTSUB, AGmax, and here. And TS_parm is a term of the parameter in corresponding with the TS_parm_CD, such as actual number of subjects and maximum age of the subjects. Next slide, please. So this slide shows you how to export our SAS dataset with XPT format. So in order to help the readers to understand the function of the code on each line, we put the number just right beside of the code. For example, the number 1 is to define two lib names. SAS file, for the SAS file is to save SAS dataset, and the next lib name is XPT file, is to save file with XPT format. We should notice that here, when we create a path for save the XPT file with export agent, we should specify the name of that XPT file. For example, at here, that XPT file's name called TS.XPT. And after that, we show two different ways of creating XPT file. So the first way is number 2. We create XPT file with data step. And the next way is use proc copy statement to create XPT file. The syntax is pretty straightforward, and you can just copy this code and type in your own software to see if you will get the same output as we put at the bottom, number 4. Next slide, please. So this slide shows you how to use a macro to generate a pile of XPT file in SAS. So in the pharmaceutical company, almost every pharmaceutical company has their own macro to generate XPT files efficiently and effectively. And here we put this macro called create XPT. For this macro, we have 2 parameters. The first parameter is inlib. So this is the name of SAS library containing the input dataset. For example, you can say inlib equals to SDTM. And the second parameter is XPTDIR. This is a folder where the XPT files will be generated. So in the bottom, this is output of XPT files generated by SAS macro. So we highly recommend you to go to read the related materials that teach you how to generate macro with XPT file. Next slide, please. So this slide is show you how to use RStudio to generate final dataset in XPT format. So first we should know that R is a programming language that developed cooperatively and non-commercially. RStudio is a commercial product. It's an integrated development environment as a tool for statistical computing and graphics. So in this section, we will use RStudio to create raw TS dataset, to create SDTM.TS dataset, and export the R file with XPT format. Next slide, please. So this slide shows you how to generate raw TS dataset in RStudio. So after we install R and RStudio, then you can use either option 1, SAS export package, or option 2, Heaven package, to read SAS dataset in RStudio. For example, here, the number 1 is install the package, SAS export. So SAS export package is to provide function to read list contents and write SAS export files. The number 2 is tell you how to invoke the library function to load it into the R session. So here we use 2 library functions. The first is SAS export library function, and the second is HMISC library function. So for the HMISC library contains many functions, such as useful data analysis. We need to use data frame function and the label function under HMISC library, which we will show you in the following slides. Then we can use library SAS7BD80 to read SAS datasets into RStudio. And there's a second way to import the SAS dataset into RStudio. When we store the package called Heaven package, then we need to invoke the HMISC library function and Heaven library function. Next slide, please. So we see at this slide, we have 2 methods to create our R file. So in the left one is we use data frame function to create R file in the RStudio. So in that condition, it means we don't have SAS dataset in our SAS. So it means we cannot import the SAS dataset from SAS into RStudio. In this condition, we should create our own R file in RStudio. So we use data frame function. You see at the left side, we combine with vector function to create values under each variable. However, if we already have SAS dataset in SAS, so the thing we need to do is just import the SAS dataset from the SAS to RStudio. Then there are some ways we put on the right side, the method 2. For the method 2, we have another 2 conditions. If you install the Heaven package, as we mentioned in the former slide, then you need to use read_SAS function to read SAS datasets into RStudio. However, if you install SAS export package from the former slide, then you need to use read.sas7bdat function to import SAS datasets into RStudio. Next slide, please. And then you need to use label function to assign the labels for each variable. When you use label function, the precondition is you already invoke the hmis function, which we already mentioned before. After that, you use write.export function to export the R file with XPT format. A syntax shown at here. And after you run this, the output will show in the bottom. Next slide, please. So this slide is a fast way to create all XPT files in RStudio. So this way is very similar as we use macro in SAS. The first process is we create a list with a list function. And the second process is we create a list of all the SAS7bdat files in the directory with a dir function. And the third process, we use for loop function to import all SAS7bdat files from SAS library to RStudio. And number 4 is concatenate the string and create the path where store all XPT files. And the fifth process is export all XPT files with write_XPT function. So for this part, it's very important in our paper, and we highly recommend you to go to read our paper very carefully, at least this part. And in the paper, we show you each output generated from each line of the code to help you understand what's the function of that part. Next slide, please. So this slide is an output window generated from the code in the former slide. Next slide, please. So this slide shows you how to use Python to generate final datasets in XPT format. So first, Python is an interpreted, object-oriented, high-level programming language. In the pharmaceutical company, there is a growing acceptance towards using Python to deal with data and to deliver the programming outputs. Said here, after we install Python, we should open the cmd.exe command line interface, then set the directory where Python is installed, and type in pipe install export, and show below. We put the red box, emphasize that part. You can follow this process and create your own Python file. Next slide, please. So the next step is you should open the IDLE application, which is an integrated development environment for Python. So this environment is for creating the new file. So in this start, we need 2 packages. The first is export, and second is pandas. For the export, which is a marginal for providing node function for read data from a SAS file. And the second is panda. Pandas is a standard for data analysis and management. Next slide, please. Next slide, please. So this slide shows you how to generate raw TS domain in Python. So we use data frame function. So this way is very similar as we use the data frame function in RStudio. But the difference is we use bracket to create those values under each variable instead of the vector function. And the number 2 show you how to use set_option function to display the complete tables. Because if you have many columns and many rows in the Python, so you might not see the complete table in the output window. That's the reason you need to use this set_option function. The number 3 show you we use head function to show whole TS dataset. So next slide, please. So this slide is show you how to export our Python file with XPT format. The syntax shown at here, so it's very straightforward. Use with open function to export the TX frame.python file with XPT format. Then we store this XPT Python file into this location. So this location is included in the first part of our syntax. Next slide, please. So this slide is the 3 TS.XPT files generated from 3 programming languages. The upper one is a TS.XPT file generated from SAS, and the middle one is a TS.XPT file generated from RStudio. And the bottom one is a TS.XPT file generated from Python. Next slide, please. So next slide, please. So after we use SAS Universal to open these 3 files, and we find there are some differences among them. So firstly, when we look at TS.XPT file generated by SAS, the length of the variables is corresponding with the standard variables length in SDTM specification document. SAS allows us to change the variables length. However, in both R and Python, the variable's length depends on the length of the elements in a vector. Secondly, when we look at the label column, both SAS and RStudio can assign variables length. However, it doesn't work in Python because the Python export modular doesn't have advanced properties capabilities, such as variable length cannot be assigned. Absolutely, we can use another function in Python to assign the labels or to do the data analysis, but it's not the main topic from our paper, so we won't talk about that. If you are interested in digging the other function of the Python, you can go to read some related papers. So the conclusion here is SAS and R include more advanced capabilities to edit XPT file. Thanks, everyone. So now I will give it back to Todd.
Thank you very much, Yuting. And thanks everyone for attending our presentation today. We hope you enjoyed it. We hope you have questions for us. Our contact information is on this slide. Again, we both work at Vertex. Feel free to reach out to us if you have any questions about our presentation, or we'd definitely be interested if you've taken this topic and kind of moved forward even more than we have with it. So again, we appreciate your attention today. We're hoping you have a great time wherever you are at the Virtual SAS Global Forum 2021, and we look forward to hopefully interacting with you when we get to the conference. Thank you very much.
Thanks. Bye.
Bye.